It has sort of a wavy features, and you see those steps, it looks like water waves or something like that. Ripples, sort of ripples on the surface. So another, just a pretty surface. And what surface does that one look like? It's glass. You see these big areas that are smooth and little smaller ones? Borosilicate. So there's a mixture of boron and silica, and they have these interesting particle arrangements. So you can look at composite structures of different kinds of atoms and elements that are together. So this is 30 by 30 microns for the whole view. And that's what glass looks like under an atomic force microscope. This is something called MOS2, moly disulfide. It's an important lubricant in industry. Whoops. And the spacing of individual moly and sulfur atoms, you can measure the spacing between different atoms, are used to calibrate the microscope. They're so regular and so well defined that you can look at surfaces and use an atomic lattice to calibrate your instrument and to know what sizes things are. This is a dendritic. It's just a pretty fractal array. It looks like trees or branches growing out. This is copper that was grown on a glass surface. And it's just another pretty image to take a look at to see what things look like. If you have something flat, you can put it on an AFM and you can see it. This one is a very highly resolved view of calcium fluoride. So the bright areas are calcium, the dark areas would be fluoride. And again, it's very regular periodic arrangement that you can see with an AFM. So let's move to part two. We saw atoms. Most of the time, you either have one or two atoms, and they're arranged in a periodic crystal regular fashion. They're all the same size. Molecules are a little bit more complicated, sometimes difficult to interpret what you're looking at. I'm only going to show a couple examples from my laboratory. This is a molecule called a self-assembled monolayer. It's actually, these are carbons in a backbone, and they stand up at a tilt. They bind to a gold surface with a sulfur gold, and they have a methyl group presented at the end. But basically, it's like a whole bunch of atoms strung together in a chain, and they stack like soldiers into a beautiful crystal. So if you were looking at the top, now we're not going to look at the side view. We're going to look at the top view of what these molecules look like when they arrange on a surface. And what they look like, the surface is going to have some bumpiness to it already, and you can only see like a circle where the methyl group is at. And this is what it looks like. These are AFM images from my lab where the surface doesn't look all that flat. It doesn't look periodic like little individual bumps when you look at it from a bigger view. As you zoom in, can we see those individual methyl groups? What you see when you zoom in, you see little dots. Those are actually dents in the gold underneath, and they're 2 angstroms deep. So what we're seeing are these what are called etch pits, or molecular vacancy islands. They're little dark spots like Swiss cheese where the atoms are missing, and they're just missing a gold atom, so the molecules look a little bit shallower. And so zooming in a little bit further and further and further, now you see the bright and dark spots. The bright spots represent where the methyl groups are. And so even a molecular lattice, if it's organized, you can see it with an AFM. So that's my example of molecules. I have some more biomolecules in a few minutes I'll show you. So that's what some pictures look like. Part 3, biological. Okay, we'll move right into that. And I have to confess, this comes from a college laboratory, a college lecture. There's some big words here. But they're biological samples of viruses, chromosomes, DNA, cells, and proteins. Basically what people and animals are made of. You know what a cell is? It's something very small. Well, cells you can see with optical microscopes, but sometimes you want to see the details of what's inside the cell. Or you want to see a protein, you can't see that with an optical microscope, but you can with an AFM. This is TMV. I like TMV's shorter. Tobacco mosaic virus. Tobacco mosaic virus. Wait a minute. Tobacco's a plant, and the mosaic virus is a virus that attacks the tobacco plant. So the cigarette growers care about this particular one. They look like little rods, little short rods. There you go, the tobacco plant. We dry it, roll it up, and smoke it, okay? And the tobacco mosaic virus, you see the dark patches on the leaves? That's the tobacco mosaic virus. And this is what it looks like schematically. It's like this helix of protein, and it's all wound together in this regular shape, these little rods like this. So this is an AFM image of TMV, tobacco mosaic virus. Interesting structure to study. These are rafts of those TMVs all clustered together. And here's an electron microscope view where you can see what they have, these rod shapes, and they assemble together on surfaces like in these beautiful little patterns. So that's an AFM images can show us what tobacco mosaic virus TMV looks like. Okay, and these are RNA molecules of the TMV virus. RNA is even smaller, but it's still on the order of tens of nanometers. So what's another thing you could look at that's inside a cell? A chromosome. Chromosomes, human, animal chromosomes. They look like sort of an X. Their dimensions are a little bit bigger. You start with the chromosome, you unwind it, it goes and it gets wound down into individual DNA fibers. We can see all that with an AFM. So here's what AFM pictures, they look like an X. These are AFM images of chromosomes. And here's DNA sort of as this strand of atoms and molecules that are wound together like this. What does it look like with AFM? Hopefully I'm showing you some things you haven't seen before. So this is what plasmid DNA, what the DNA strands, they're like ropes that are just sort of tossed on the surface, and they make tangles and they twist together like braids or like spaghetti or pasta all wrapped around. That's what it looks like with an AFM. So that's a visualization of supercoiled DNA with an atomic force microscope. So this is the structure theoretically that we've always thought it had from the Watson-Crick movie about how it was invented. This is what it looks like with AFM. And you can color your AFM images black and white or with different colors. Here's some strands of mitochondrial and plasmid DNA. These are from different types of organisms, and they look like ropes that are sort of randomly arranged on the surface. And here's another one, DNA silane condensates. So what this image from the college lecture is, they bind some molecules to the DNA. And we're trying to study how those molecules bind to DNA. And so you see the little bright red spots. When they bind, you can see where the red spots are. You can actually see them with you can see molecules bind to each other. You can study chemical and biochemical reactions with an atomic force microscope. You can see where they bind. It's a different way of thinking about science, to actually be able to see the chemical reaction. Here's some plasmid DNAs are in a circle. So whenever you see a circle and they call it DNA, you know that that's a plasmid. Now this is a model of something called ferritin. Ferritin's a protein that every animal and plant has, and ferritin means iron, and ferritin has to do with binding the iron, and why everybody needs a little bit of iron in your body, but if there's too much, ferritin takes care of it. Ferritin regulates how much iron to keep your iron in balance in your bodies and in your cells. They're little spheres, and look, with AFM, you can see these little spheres of ferritin. Every single one of these spheres is an individual protein, an individual molecule of ferritin. So you can see things that are really small. They're only about 10 nanometers in size, to give you an idea of what size. Biomolecules are usually pretty big, so they're something you can see pretty routinely with AFM. Atoms, you move down to